Buenos tardes, and thank you all for staying till the bitter end. In the spring of 1961, President John F. Kennedy announced an audacious plan to land a man on the moon in a decade. When I was a young boy growing up, I watched the Mercury missions, the Gemini missions, the Apollo missions, and that all led to an amazing day in the summer of 1969 when Apollo 11 landed a man on the moon and Neil Armstrong uttered those words, "One small step for man and one giant leap for mankind." For me, this is what it meant to be. This is the kind of species we want it to be, the theme of this conference. What kind of species do we want to be? A species that explores and understands the universe while at the same time understanding and appreciating the fragility of life on our small little planet on the edge of the Milky Way. And since that time, a moonshot has come to mean a bold endeavor to attain a nearly impossible task. Well, the first moonshot in biology was the Human Genome Project. The Human Genome Project was launched in 1990. It took 13 years to complete. It involved the activities of hundreds of scientists in more than 20 institutions around the world. The project cost $3 billion at the time it was conceived. That would be in 1993 dollars. And in a 2013 report done by Battelle, a private foundation, looking at the impact of the Human Genome Project on the US economy, it was found that the Human Genome Project generated more than $1 trillion in economic activity for the US economy. By any measure, even the most conservative measures, the return on investment of the Human Genome Project was more than 140 to 1. And it's not just the return on the investment, right? It's not just the money. The Human Genome Project revolutionized our understanding of biology, and it also led to an entirely new field of medicine, which we've heard about over the past week, called personalized medicine, or sometimes precision medicine. In addition, the technologies that were spawned by the Human Genome Project spilled over into other areas of science, particularly in agriculture, where the cow's genome was sequenced, the pig, the chicken, and numerous crops and species of agricultural importance. And the results of those projects led to a complete transformation in the breeding industries for plants and for livestock. So much has been learned. So much in the last 25 years has been learned as a result of that initial project that was started in 1990. But an amazing fact is that all that we know in terms of the genomic biology of life on our planet is derived from the sequencing of less than 0.2% of the known eukaryotic or complex life on our planet. And so we began to ask ourselves the question, what could be learned from the other 99.8% of the organisms on our planet? And that led to the creation of what we now call the Earth Biogenome Project, which is the next moonshot for biology. The Earth Biogenome Project is a grand challenge to sequence all known eukaryotic life, that's 1.5 million eukaryotic species, in a 10-year time frame. And the purpose for doing that is to create a new foundation for biology and to create solutions for preserving biodiversity and for sustaining human societies. Basically, an infrastructure for the new biology and a new bio-based economy. Now, why do this? Why now? Why is this important? Well, we know the Living Planet Index tells us that 52% of the vertebrate population on the planet Earth has been declined over the past 40 years. We also know from the International Union for the Conservation of Nature that of the 80,000 species of plants and animals that have been surveyed, 23,000 are threatened and endangered. We know that extinction rates are happening at greater than 100 times the background rate. And quite honestly, we don't know the consequences for the future. As we lose biodiversity at this rate, and perhaps up to 50% of all species on Earth today may be gone by the end of the century. So we are in the midst of the sixth great extinction of life on our planet. And if we look at this picture, you can see that the Great Barrier Reef off the coast of Australia is one ecosystem that has reached its tipping point. And we really don't know the consequences. We don't know where the tipping point is for the threat to many ecosystems on our planet that are critical to our very own survival. And so, one of the things that's very important in the face of this devastating extinction of biodiversity on our planet is to create what we've called a digital library of life on Earth. And that is certainly one of our goals. This digital library will basically capture the collective intelligence of 3.5 billion years of evolution of life on our planet. This is not just my idea. I have worked over the past 3 years with 23 distinguished genome scientists around the world to develop a detailed plan for the Earth Biogenome Project, which was published in 2017. And this particular paper and the subsequent activities that we've had in talking about the project has generated enormous public interest in what we're doing, as well as great interest in the scientific community. As I said, public interest has been large. After the paper was published, the Times of London covered on their editorial page with this title to an editorial, which is "Mapping the Genome of Every Living Thing Will Revolutionize Biology." And they said in the article, the world should grasp this opportunity to see what lies in the code of our fellow creatures. They also said, as an astoundingly audacious plan, and the most astonishing part is that it's achievable. Now, I'm not sure how the Times of London knew that this project was achievable, but in the next slide or two, I'm going to try and convince you that this is the case. We already know that the existing DNA sequencing technology can do the job. We need no further increases in technology actually to do this in 10 years. What we need is enough machines and enough expertise in the world to get the job done. What's it going to cost? We estimate the cost to be $4.7 billion over a 10-year period. And that includes everything. That's not just the sequencing cost. That's the cost of collecting and procuring the specimens, preparing the DNA, sequencing the DNA, assembling the DNA, assembling the sequence, annotating the sequence, and analyzing the sequence. So it's a complete package. Let's compare that to the cost of sequencing the human genome in 1993 dollars, which, as I mentioned, was about $3 billion if you add it all together. $3 billion in 1993 is $5 billion today. So if it's one message I'd like you to take home today, it's that the cost of sequencing 1.5 million eukaryotic organisms will come in at less than the cost of sequencing the human genome just 20 or so years ago. And that's amazing. You say, well, still $5 billion is a lot of money. Biologists aren't used to that kind of money. Well, let's take a look at some of the other projects. We have some physicists, very notable physicists in the audience. I'll point out the Large Hadron Collider. The Large Hadron Collider in Switzerland produced a couple of years ago a confirmation and discovery of the Higgs boson. For that to happen, that cost $13.2 billion. And there's an operating budget for the Large Hadron Collider of $1 billion every year. And I mean no disrespect to my colleagues, high-energy physicists in the audience. I'm just trying to draw a comparison that says, ask the question, is the value that we get from confirming, discovering the Higgs boson any more than the value that we will get from sequencing 1.5 million known eukaryotes? And so, how will we go about doing this? Well, we've established a global network of networks. This is just a model, not meant to be comprehensive, of all of the organizations and laboratories around the world that are participating. But this is a global network of networks of laboratories that have expertise in high-throughput sequencing, DNA assembly, annotation, as well as participating scientific communities that are focused on specific taxa. For example, the Vertebrate Genome Project, which is designed to sequence all 66,000 known vertebrates. There are projects related to insects, there are projects related to fungi, there are projects related to plants. And all of these projects have come together under the umbrella of the Earth Biogenome Project. And all of them, when they committed to the project, they signed a memorandum of understanding that says that everything, all the sequence information that is generated to the project will be made freely and openly available to everyone in the world. Not only that, you commit by signing the memorandum of compliance with the Convention on Biological Diversity and the Nagoya Protocol on Access and Benefit Sharing, which basically says if there's any economic benefit that derives from a particular organism, your country and even possibly local indigenous populations will also get to share in that benefit. So today, as we stand, we have 24 institutions around the world in 10 countries that have made this commitment. And we are currently all working together on implementing the plan for the project. So, sorry for this slide, but it's a little technical, but it does make the point of how we're going to go about doing this. There are two strategies that we're going to implement. One, which we call the phylogenetic wave, or the phylogenomic wave. As we work down through the classic Linnaean taxonomy that describes life on the planet, we start at the highest level. There are 3 domains in the Wozian tree of life: the eubacteria, the archaea, and the eukarya. And there are representatives already for those 3 known life domains. We work our way down, and phase 1 of the Earth Biogenome Project is designed to sequence to the taxonomic family level approximately 9,000, that's the total number of eukaryotic families that exist on Earth within the first 3 years. It's not so bad. That corresponds to about 65 genomes a week. We anticipate 10 labs, so it's about 6 genomes a week, and this is totally within the realm of feasibility. In years 4 through 7, we're going to go to about 180,000, a representative species of all the genera. And in the final phase of the project, the remaining 1.4 to 1.5 million species, everything that we can get our hands on. And it's important to note that although our target is all known eukaryotes, 1.5 million, we only know approximately 10% of what's there today. Only 10%. So in reality, what's been sequenced today, the 3,000 genomes that have been sequenced to completion, represents less than 0.1% of the biodiversity on the planet. So that's strategy one. The second strategy is one that I think is very exciting and has got many governments around the world quite interested in the project. It's an ecosystem sampling. That is, to sequence very deeply at a particular site. Could be a mine that has had a severe environmental degradation. It could be the Atacama. It could be the rainforest. It could be any place on the planet, particularly those areas which we call biodiversity hotspots, where we have large numbers of endemic species and also a great loss of biodiversity in those areas. There are 36 of those around the world. So we're talking about very deep location sampling in a selected number of ecosystems around the world. And importantly, we've heard a lot about climate change, but not very many solutions that can be implemented to save some of the most important species that we have that are providing ecosystem services to maintain our climate, to maintain our food supply. And so having the genome sequences provides us with an enabling technology that will allow us to monitor biodiversity in these hotspots over time. Everything that's there, to understand how genetic changes, how adaptation happens in the face of environmental change. And finally, all of this, as we monitor in detail ecosystems, will allow us to produce a multidimensional view of life on Earth. So let's talk about the tree of life and the kinds of things that we already know, the kinds of benefits that we've already derived from nature. Just imagine what we'll discover from 1.5 million additional species. Difficult slide to see, but I wanted to make the point. First goal is to revise and reinvigorate our understanding of biology and evolution. How did eukaryotic cells arise? How did multicellularity in eukaryotes arise? What are the rules of life? These are the fundamental questions that can be asked. I don't know if you can see it. If you look, there's a small red dot. This is not the tree of life, but this is the treetop of life. And all of eukaryotic life is now characterized using molecular phylogenetic techniques that tells us that eukaryotic life can be divided into 5 major subgroups. Doesn't it remind you of the pictures you've seen that you are here, when you see the galaxy and we're just this little planet, Earth at the edge of the Milky Way galaxy? Just look at that. We're more closely related to the fungi than we are to many other types of animals on the planet. That's what molecular genetics is telling us. This is very much in what Richard Dawkins was saying this morning. We are just another organism on the planet, and we have a difference in that we have brains, and we have the ability to do things that affect all other life on our planet. So that's first. Second, this is what I do in the lab and what my group, and what we're trying to do is understand how eukaryotic genomes, not genes, as Dr. Dawkins talked about this morning, but whole genomes evolve, how chromosomes evolve, and how chromosomes rearrange over time, and what their role is in adaptation and speciation. And so this is what led me into proposing the Earth biogenome, because I had questions that I could not answer until we get them all. So that's first. Second, maximizing returns to society and human welfare. A number of things that are going to benefit by having these organisms. First, of course, we can generate, once you have the blueprints, you have biological blueprints for generating new biomaterials. You have the ability to create new sources of renewable energy, new sources of drugs to treat inherited and infectious diseases, identify drugs to slow aging, and new genetic resources to feed a burgeoning world population. So is this pie in the sky? No, it's not. There are very real examples. 80% of the drugs that are used on the planet today derive from natural resources. But very few, less than 200 plants of the 390,000 known have been sequenced. We have drugs for treating malaria, for treating pain, for treating cancer, for treating inflammation, for treating blood disorders, all derived from natural products, from plants. There are 20,000 species of marine invertebrates, less than 50 have been sequenced. There are some notable compounds that have been derived for treating cancer from marine invertebrates. Those are given here, as well as the first drug for treating HIV. What else? Those were plants, marine invertebrates. How about the fungi? More than 5 million species of fungi. The statins that many of you, I'm sure, in this room are using to control your blood cholesterol levels were first identified in a mold that grew on an orange. Much more to discover in the fungi. And of course, in agriculture, this is one of the most important things and areas of effort, is that so few species are used to feed the human population. Of those 360,000 or so 80,000 plants, only 12 species provide 75% of the total food supply, and most of that is wheat, rice, corn, and potatoes, which is more than everything else combined. The same is true for livestock. So the project will be extremely important for generating new sources of plant-based protein. Dr. Oliveira talked on Monday about the importance of finding substitutes for animal protein to preserve the environment. New sources of protein and new sources of genetic variation to improve disease resistance and drought tolerance and other agronomically important traits that will allow our agriculturally important crops to survive the changes of environment that are coming in the next 50, 75 years. And finally, as I mentioned in the beginning, conservation, protection, and regeneration of biodiversity. It's critical that we understand how to protect ecosystems and how to manage ecosystems. And today there are programs throughout the world that are aimed at doing this that are based on genome sequence information. I put just a few of the notable examples here. The Tasmanian devil in Australia is being managed genetically. The African cheetah, the California condor, these are all examples of successes that have been achieved, at least in part, due to understanding the genomic information in these species. And it may eventually be possible. It's not possible today, but scientists are at least talking about it. They're talking about de-extinction by having that digital library of life on Earth. Perhaps someday we can bring back the mammoth or the thylacine, the Tasmanian tiger. It's not a tiger at all. What it is a large carnivorous marsupial, okay? The last one died in a zoo in Tasmania in 1936, but in the Australia Museum there are lots of samples of this species in the drawers. Maybe de-extinction will be possible with this kind of information in the future. And so, where are we with the project? The project officially launched in London on November 1st, 2018 at the Wellcome Trust. The Wellcome Trust sponsored this event, and at the event, the Sanger Center announced their contribution. The Sanger Center is the biggest genome center in the United Kingdom and contributed a significant part to sequencing the human genome. And they have made a commitment, a firm commitment, over a 10-year period to establish a program to sequence all eukaryotes in Great Britain, in the UK, 66,000 species. Similar efforts are underway in Norway, in Australia, in California, in other countries around the world, and this is just the beginning. And the beginning must include Chile. Chile is a very important country. It has a lot of biodiversity, more than 4,000 plant species, more than 600 vertebrate species, more than 1,000 marine mollusks, and 50% of the known species in Chile are threatened or endangered. So Chile should be involved, and indeed they are involved. I'll talk to you about that in a moment. And last September in New York, the first of those 9,000 reference genomes was released to the public by the Vertebrate Genomes Project. Sequences for all of the classes of mammals, at least one representative, 4 mammals, 3 birds, 1 reptile, 1 amphibian, and 3 fishes. Now, 14 doesn't sound like a lot, but the quality of these 14 sequences that were completed in a very short time period exceed by a long shot the quality of the human genome sequence that was released in 2003. And the Wellcome Trust Sanger Center has released the sequences of 25 iconic species of Great Britain for the 25th, in celebration of the 25th anniversary of the Sanger Center. And I'm very proud to tell you that this work is also going on in Chile. We heard about this on the first day from Florencia. The 1,000 Genomes Project. There may be 1,000 human genomes and 1,000 species drawn from the biodiversity of Chile. The project is just getting underway, but there is a funding commitment to it, and Professor Miguel Allende is leading this project here in Chile. Now, this is not to say at all that we don't face challenges. We have a long uphill climb. Getting 1.5 million vouchered specimens, that's the problem, not the sequencing, it's acquisition and distribution of the specimens. So we need innovation. We're talking about drone technology with robotic devices that can go and collect species from remote and hostile areas of the world. Even sequencers on board those robotic devices to provide positive identification. And of course, we've talked nothing about the data. This is exascale computing. We have experts as part of the Earth Biogenome Project who are dealing with this question. We have a very, very big data problem. And so that's what we're trying to accomplish. And, you know, nobody is saying that this is going to be easy. In fact, John F. Kennedy, when he launched the Moonshot to land the man on the moon, said, "We will do it not because it's easy, but because it's hard." And so the Earth Biogenome Project has the capability and the capacity to tell us who we are, where we have come from evolutionarily. And it also can tell us the kind of species that we don't always want to be. Some of the things I think that we'd like to draw from nature, some of the lessons would be, for example, the ants. There are species that are ants that are totally altruistic. They give their life for any other ant to save the queen. Elephants can live without cancer their entire lives, almost a zero incidence of cancer. And there are species of turtles that can live for 250 years. But in the end, I think, given a choice, none of you would want to be ants or turtles, maybe not even elephants. I think our best choice is to be human and to use our brains to understand the living world and to make the world a better place for future generations. And so, for the future, we want to change and not think about going forth and conquering, which is the way we did things in the past, but to go forth and sequence. That's the job we have before us. Thank you very much.